So first of all, I would like to thank the local group committee for asking me to speak today. Really, it is on what I've been doing, or the talks based on what I've been doing on my PhD, which is looking at really fractal measures for the early detection of glaucoma, and then as well a spatial model uh, that can be used in conjunction with change point detection to sort of analyze the retina to see where um, potential areas where there's density loss of cells occur, and then ultimately leads to your loss of vision in the disease. So first of all, what exactly is glaucoma? So it's a neurodegenerative process, and it is caused by damage to your optic nerve, an optic nerve head. So at the back of the retina here, indicated by the arrow. This damage then leads to a loss of the retinal ganglion cells. So these cells transmit visual information from your eye to the brain. And the way it works is if you were to look at an object or an image, so for example, a light bulb, your retinal ganglion cells will take this light and transmit it into electrical signals. They then bundle together here around the optic nerve head and then transfer along the optic nerve to the brain, where it's then transferred back into an actual image. So um, what exactly is the issue? So one of the problems, or what I've learned really over the past few years, is it's a very complex disease. So really what happens is, as I said, it's a neurodegenerative process. So what's happening is there's two steps: primary retinal degeneration and secondary degeneration. And in this sort of primary stage, what's happening is there's an insult caused to the optic nerve head, and this is then leading to cell loss as a result of this injury. But then secondary degeneration is linked to this sort of primary degeneration, because the primary degeneration, the cells that are lost, they will cause an inflammatory response in the area. This will then lead to the area surrounding the cell to become toxic. So neighboring cells essentially will succumb to this toxicity and will eventually die as well. What can happen, or sorry, the therapeutic targets that treat the disease at present, sometimes they do work and sometimes they don't, because they're targeting this secondary degenerative process. And sometimes they're intervening, and when they're intervening, it's actually too late. The process has already begun, and it's hard to sort of come back from that once it's started to begin its process. So really, an early detection of the disease is essential. And at present, the way you go about detecting glaucoma is you'll go to your optician. They will then refer you to a specialist if they think you have glaucoma. So they'll perform another number of tests there and see whether or not you may or may not have it. If they think you do, they refer you to the specialist. The specialist then analyzes you for about 6 to 12 months. And over that period, then if they think you have glaucoma, they'll recommend a treatment. But as I said, sometimes the treatments don't work. So then it takes another 6 to 12 months for them to analyze you again to see whether or not the treatment is working for you. If it's not, you go back to the start again, really, and you get another treatment, they analyze you again. So it can become quite a lengthy process, and by which stage you actually have lost or have had substantial damage to your to the retina has already been done. Another issue that we have is the high plasticity of the central nervous system. So essentially, your body has a mechanism to cope essentially when things are going wrong. So what this means is you will lose approximately 80% of your retinal ganglion cells before you would even begin to develop symptoms of the disease. So what this sort of looks like in the eyes of someone with glaucoma. So because of the central high plasticity of the central nervous system, you could go to sleep with normal vision, and then you could end up waking up with slight loss of peripheral vision. This will then continue to worsen if there's no treatment, or there's no treatment intervening there. So, and as well, also, you can then start to lose your central vision as well in spots. So it ultimately leads to blindness, and that's why sort of early detection of the disease is essential. Now, we can't analyze human retina for obvious reasons, because we can't image them. And even if we could, I don't think anyone would be too keen to let you watch the disease process happen rather than actually helping you out. So we have retinal hole mounts of mice, and these mice have been subject to different medical procedures, or as they're referred to as biological models. So if I do say model in this context, sometimes it will probably mean the biology or the procedure that has occurred in order to simulate the disease process. These are an example of the retinal hole mounts on the slide. So they have been dissected and flat mounted, and then I've shown as well a small subregion of the retina, just to give an idea of what the pattern looks like of the cells. We also have then information about the actual cell size itself, as well as the optic nerve head, where its location is, which is approximately in the center there of that hole mount in the white circle. But why we use mice is essentially because this is the closest animal that we can get to human retina. Their density and structure is closely, and the way the cells are lost is actually closely related to how it's lost in humans. So the data set that we're working with contains a total of 61 retina. So there was, as I said, different procedures. Each mice underwent different procedures. So there was a cohort of 15. They were in the control group. So essentially they didn't undergo any process. So they were the healthy retina as such to compare to. Then there is a model or a procedure known as the partial optic nerve transection. And there's a total of 24 retina within this group. So just to sort of give you an idea of what that medical procedure looks like. Essentially, the mice are undergoing surgery, and then they're exposing the optic nerve and then making a small incision, enough to do damage, but not enough to completely cut off the vision from the optic nerve to the brain. So the idea behind it is it's essentially a more severe model of the disease. So the cells be lost quite rapidly throughout the disease process. And because of that, we have different dissection times, which we group the patterns into as well. So we have 4 dissection times of 3, 7, 21, and 56 days. The other model that we used is the ocular hypertension model. This model has a total of 22 retina within its cohort, and it's actually probably closer to what would be observed in human or glaucoma. So this procedure in particular, because essentially what happens in glaucoma a lot of the times is the part of your eye which controls the fluid pressure within it can become blocked. And then what happens is there's an increased pressure, and this increased pressure causes damage to your optic nerve. And this then leads to the retinal ganglion cells being lost. So this procedure is essentially to simulate that type of loss. And within it, because it's actually a more milder model of the disease, so our dissection times are slightly longer in some cases. So we have 7, 21, 56, and 84 days for our retina within that group. So really, what we wanted to ask, or what we wanted to achieve from this data set, was firstly, if we could develop methods to help with the early detection of the disease, but then as well, if we could develop a mathematical model to look at areas which are at risk of high incidence of the disease. So the reason why I want to do this is because there's been recent advances in adaptive optics, which allow clinicians to visualize a small subregion of the retinal ganglion cells. So inferring sort of where they should be looking, and also what methods they could be using to help with the detection of the disease earlier. So focusing on the first question, first of all, really, there has been previous studies that used this data set, and they looked at distance-based measures like our nearest neighbour distance, regularity index, which was defined as the average nearest neighbour distance divided by its standard deviation, and then also the average cell size as well. And what they found was that for our PONT group, the more severe disease, these methods perform quite well, but for more milder model of glaucoma, these procedures didn't actually perform as well at detecting the disease, or they were quite poor. So this led us to look at fractals. I probably wouldn't look too long at that image, or it can mess with your head a wee bit. But essentially, when people think of fractals, they think of this sort of object here, which is sort of self-repeating or self-similar, infinitely repeating image. But point patterns as well can be fractal. And that is why we went down the route. But as well, we also have that our retinal ganglion cells are self-similar in structure. So there have been papers published which look at one of the measures that I'll talk about in a second, known as the fractal dimension, and it applied in the context of retinal ganglion cells to determine the complexity. But as well as that, then, because look, sorry. But then one of the other interesting, or one of the other sort of reasons why we like to use fractals is it's closely related to point patterns in some contexts. So we have that our fractal dimension is one of the measures. So this measure looks at the complexity of your pattern. Really, it stands really for a fraction of dimension. So if you think of a square, it's dimension 2. Whereas if you were looking at a coastline or something more complex, if you pass it off as dimension 2, you wouldn't capture the complexity of that, the intricate details of it. So really what the fractal dimension is doing is looking at the complexity of our pattern. So as I said, the K function is widely used in spatial statistics as a summary measure. The way it's calculated is for your point pattern, you will select a point, you draw a circle of radius r around it and count the number of points that fall within this circle. And then you repeat the procedure for each point in the pattern and take the average. You then multiply it by an estimate of our intensity, which is given by the area of our study region divided by the number of points. And then for the fractal dimension, it's simply taken from the gradient of a graph of our plot of K with increasing value of R. The only issue with this is it only seems to work for when we have distinct clusters in our patterns. So if we have the patterns are randomly distributed, or the points are randomly distributed, or if they are distributed quite regularly throughout the study region, we find that they estimate the fractal dimension quite the same. So the standard method then that is used is actually a more simpler one. It's the box counting algorithm. So what happens in this context is essentially you overlay a square grid across your study region. You're interested in whether or not your box contains a point, not how many points are contained in the box, just whether or not there is a point.
Can people see that? You square, you see squares there? Oh, it seems very big to me. Sorry.
No, you're okay. So we want to know whether or not the box contains a point. And then what we do is we repeat the procedure for different box lengths R, and then we get a plot of the number of box containing points against the number of, or the different distance, or the different box lengths. And again, it's calculated through the gradient of this graph then. So this is the more commonly used method for detecting or for calculating the fractal dimension. And what the issue for us with this measure is that because we're only interested in boxes with points, when it comes to more milder models of the disease, they're more random and it's not as noticeable where the points are being lost. So this measure may fail in terms of detection of it. So that's why we decided to look more into the detail of more or less commonly used measures of fractals. So in particular, the first one was the lacunarity. So the lacunarity is a measure of the gap distribution. Essentially, the fractal dimension looked at the complexity. The lacunarity now looks at how these points are distributed within the study region. Are they clumped together in a cluster? Is there large space between these clusters, or is there very little space between the clusters, or are there random points throughout the region? So the lacunarity is capturing this this sort of distribution of the points as well. So this is an example of where lacunarity might be useful. So these two patterns were generated through two different processes. One was a cluster process, the other was a homogeneous Poisson process. The patterns are arranged differently, and we actually obtain the same value for the fractal dimension, whereas if we use the lacunarity, we find that they are indeed different. The way it's calculated is similar to before. It's using a gliding box algorithm. So this time we are interested in how many points are contained in our box. So essentially, we place a box, and then we move it along, count the number of points in our box of length R. This would be our box mass S. And then we repeat the procedure until the whole study region is covered then. Once we've done that, we can then get a probability distribution for our different box masses. That is the number of points in our boxes of length R. And then from this, obtain a first and second moment. The linearity is then calculated by dividing the first or the second moment by the square of the first moment. And then we just repeat this for different values of R. And again, similar to before, you'll take the gradient of the graph to get an overall value of the linearity of the pattern. The final measure is related to the theory of percolation. So it's not as commonly used as the other two. There's been very few applications where this measure has been used. Essentially, if you convert your pattern to an image, really what you're looking at is how connected is the empty space from one side to another side of your image. So if we converted the pattern to a pixel image, you can think of it as if you poured water in at the top, how far could the water travel before it stops? And what we have then is that these sites will be occupied with probability P. They're unoccupied or occupied with fluid, that is, with probability P, and then unoccupied with probability 1 minus P. And there's a value then of P where this image will connect from one side of the image to the other. And that's what essentially the figure is showing. So in A, we have that our P, our value, is less than the infinite cluster, which is referred to as the probability where these two sites come connected. And then at P, we have then, or B is the example of where we have just an infinite cluster has occurred, or our value of P is equal to P infinity. And then once we go past that value, the cluster gets bigger. And what also is appealing about this is from this image, they actually become quite self-similar, or the clusters become quite self-similar around that P infinity mark. So if you want to take it further, you can then look at the fractal dimension and apply that K function method that I looked at earlier. So the measure to get the value of P, we use a measure known as the circularity. So it indicates the capacity of flow through an object. So the first step is really to convert your pattern to a pixel image. Black points represent where there are points or whether there is an object essentially obstructing the capacity of flow. And you imagine pouring the fluid in for each of the different directions within your pixel image. So from the top going down, the bottom going up, from right to left and left to right. And this is in B. For this pattern is what the resulting images will look like. So there can be 4 different images, but then what we do is we work out the occupation percentage so we'll overlay a square grid. This square grid then we'll work out the occupation percentage, that is the number of blue pixels in this case out of the total number of pixels within that box. And then what we do is we apply a pressure field to ensure that our value is dimensionless. So for our first 2 images, this pressure decreases in the downward direction or increases in the downward direction. And for the last 2, the pressure will increase from left to right. And then to calculate our circularity, essentially we're taking the occupation percentage for each box, multiplying it by the pressure, and the pressure applied is taken from the centroid of that box. And then we divide it by the total pressure applied to the whole image. So essentially we wanted to see how well these measures compared to the previously used measures. So as I said, what previously had been done had been found to be very good for our severe model, but for the more milder model of disease, they've been performed quite poorly. So the algorithm we apply, or the way we assess this, is we use the bootstrapping algorithm. So the way it works is we essentially, for the control group to start with, we select a retina at random with replacement. We then, We then select a distance from the optic nerve head. So this distance we have going from 300 to 2400, essentially to cover as much of the retina as possible for as many of the retina. And then we need to check as well that our study region is actually in the retina, because obviously because it's the whole mount, they can sometimes have it half in, half out. So we need to check that firstly that the pattern is actually in our study region, and if it's not, we'll repeat the process. The same thing occurs if we don't have enough points in our pattern. And then what we did is, once we were satisfied with that, we calculated our measures, so our nearest neighbour distance, and then our regularity index and average cell size, which we were using in the previous studies, and then our fractal dimension, which we used the box counting algorithm because there's no clear clusters in the process. So we felt this had the best chance of showing a difference if there was one. And then lacunarity and circularity as well. And then the procedure is then repeated for the different groups as well. So for your OHT or PONT group. So firstly, what we find is for our PONT group, what we did was we plotted our ROC curves and obtained an AUC value for these curves. And this is what we've plotted at the different distances from the retina. For the PONT model, because it's severe cases of disease, they all perform very well because it is severe, really. The only thing to note is that our average cell size was the only one that didn't have an AUC value of 0.9 in any of the regions. The reason is just because of the degenerative process that happens. So your cells will grow in size when they're after the initial impact of the primary degeneration. So this is what the measure is picking up, really. What's more interesting, though, is what happens at our OHT model. So again, our average cell size performs poorly. Our fractal dimension also performs poorly. This is due to, again, the same reasons as before. So the points are being lost subtly across the study region, so it's not able to pick up this sort of change in the pattern. And then we have our circularity and lacunarity perform particularly well in our central retina region. And then our nearest neighbour distance actually did quite well, which is not what we had expected to see. But when we looked at our regularity index, we found that it didn't perform as well, which may suggest that it was influenced by larger values, but I'll look at that a bit later on. So then we took the three best measures and we looked at the central retina and we split it. And we looked then at the different dissection times. So repeating the procedure, but this time we're splitting them based on what time they were dissected in the retina. And we wanted to see how they compared in terms of early detection of the disease. And we found that those three measures, your lacunarity, circularity, and nearest neighbour distance, performed very well. They all recorded values of average value of 0.8 within those central retina or above from the 7-day mark for the OHT model. So it indicated that for our fractal measures, they have done particularly well. But then essentially we wanted to see essentially wanted to see what the influence of noise would be on our data set, because one of the issues that we have is because of the way it's imaged, our points might not actually be where we think they are. So we need to incorporate noise into our pattern to see how well these measures, although we think they're doing well, how they would do if we added noise to them. So this is what this graph is representing. So we added a Poisson noise to our pattern with varying degrees. So to compare it, we calculated the dice value or dice coefficient. So essentially, how similar are the two patterns, really? So if it's a value of 1, they're completely different, but if they're a value of 0, they're the exact same pattern. And what we found was that our fractal measures can remain consistent throughout the different values that we used, which was to be expected, given that fractals generally, what happens is if you know the value at one part of your study region, the value should be fairly similar within that sort of area as well, no matter where you take it from. Then we also wanted to see the impact of noise when we randomly thinned the process. So this time what we did was essentially we took our pattern and we randomly took out points, and then we again calculated the dice coefficient, so that the probability that a point was selected was higher for those dice coefficients that are much higher. What we see again is that generally our fractals do remain fairly consistent, whereas for our nearest neighbour distance, there actually was a significant effect noticed from that lower dice value compared to the higher values that were recorded as well. Just sort of, or just for out of curiosity, we wanted to see, so we knew that our circularity had done quite well. We wanted to see how it done whenever we took those images that were produced when we were calculating it and applied our fractal dimension to it. And what we actually found, so we did this through the K function, unlike before. So we were looking at the K function this time rather than the box counting algorithm. And what we found this time was that they actually performed not that great. But the reason is because really in and around that sort of value of P infinity, so you have an infinite cluster, that is the one side, the clusters connecting from one side to the other. Really, because it's more milder, what's happening is the points, there's only a slight change in points, and we're only increasing the density of the cluster only very so slightly, which is not really being picked up through this measure, essentially. But it was worth a try anyway to see what happened. So then, using this, essentially what we wanted to look at then was if we could develop a model that could identify areas that were more at risk of disease than other areas. The idea behind this was so that if we could model sort of the different areas at risk at different time points, then we could see where best to infer clinicians to look at, and then also to tell them essentially what it should tell them what stage the process is at, so that they should know what best treatment can be given to the patient. So trying to cut out the number of times you have to be given a different treatment, essentially. So to do this, we looked at different models, but we first of all looked at some summary statistics. So this is the calculation of our L function just for a typical pattern in our retinas data set. And what we find is that, so we have a plot, so L is given by the square root of our K function from earlier, and then we plotted L minus R against their different values of R. And what we find is that we have R values below 0 at the start, and then R values above 0 afterward at later or greater values of R, sorry. So what exactly does this mean? Well, essentially, if you have an R value or L minus R value above 0, it would indicate that there is an attraction between your points. So they're interacting with each other and they're essentially will form an aggregated pattern. And what we can do in that case would be to apply a model like a log Gaussian Cox process. But as well, you could have then that if your L minus R value was below 0, this would then indicate that you have some form of repulsion between the points. And for this type of model, you would apply something like a Strauss process. But for our data, this is an example of a small subpattern for our retina data set. We have both going on. We have repulsion between the points, but then as we get further out, we see that there is an overall aggregation. So essentially, we need to combine both a Strauss process and a log Gaussian Cox process, really to have a model that will model both the both the repulsion and the aggregation. So work has already been done in sort of this area, but it's only been recently that the density of this type of process has been appeared in the literature. So this is what's known as the log Gaussian Cox Strauss process. Essentially, we have our log Gaussian Cox process, which is given by our exponential of our Gaussian random field Z. This is influencing our intensity, so it's influencing our aggregation between the points. But then we also have a repulsion that we need to take for account for. So our cells will have physical size, and as well there will also be sort of an attraction at between sort of different veins and everything in the pattern. So this is where our gamma and our R are estimating really. And then we have a normalization constant as well. So in order to obtain what we want to obtain is parameter estimates theta. So our mu, our sigma squared, and our S are parameters from our Gaussian random field. So they are the parameters used from it. And then we also want to obtain parameters for our Strauss processor R and our gamma. The issue with it is the likelihood is, unlike other process or other densities in the literature, the likelihood isn't expressible in closed form, which means that we can't use pseudo likelihood or likelihood based inferences. We have to do an approximate Bayesian computation algorithm instead. So how this actually works is, before looking at that, I need to sort of explain how this model is simulated. So really what we do is we take realization of our Gaussian random field, and then we apply a birth, death, metropolis, Hastings algorithm. So essentially, there's a probability of half that you will have a birth, and then otherwise you will have a death. And if you have a birth, then a point will be generated with the probability density proportional to your exponential of your Gaussian random field. And then otherwise, we'll select a death point using the uniform distribution from the current pattern. So this is the approximate Bayesian computation algorithm. It probably looks quite intense at the minute, but essentially what we're doing is we have our observed point pattern, and then we can simulate patterns that will be a realization of our log Gaussian Cox process. And then what we can do is, for the approximate Bayesian computation algorithm, we select prior distributions, prior values from our prior distributions for theta. Our prior distributions were non-informative, and we based them essentially on the number of points for our mu and sigma squared and our S, and then as well what we looked at the summary statistics for our R and our gamma. And then once we had this, we could simulate our log Gaussian Cox process. And then essentially what you do is you see how close the two patterns are based on some summary statistics. Now, in the literature of the log Gaussian Cox Strauss process, they mention that there's limitless summary statistics that you can use because there is no sufficient statistic for the process. So as well as looking at the number of points and our L function, which we only evaluated at 20 points just so that we weren't overfitting the model, we also did look at our circularity and our lacunarity, so also taking into account the distribution of the points throughout the study region as well, or how they're distributed. You then check these summary statistics. If they're less than a specified distance between the observed pattern, you accept them and they form part of your posterior distribution. How we determine that distance, we use a pilot run, which is where our K-pilot's coming from. Essentially, during that pilot run, you will fit a linear model to your posterior means, and then we determine the distances based off that. So it's really the one percentile of the distance between each of the patterns and the observed pattern as well, or in that pilot run. So what we did was we applied similar technique. We looked at sort of small subregions because that is what the clinicians will be able to see. So we looked at the different areas of the retina and we've obtained different parameter estimates. So essentially for our mu and our sigma squared, we see that there's increasing as we go further out of the retina, which would correspond to the increase in density that we would see in the number of cells. And then as well, we also have that our gamma is close to 0, but this is being driven by the fact that our cells have a physical size, so there'll be a distance where no other cell can inhibit really. So that's why we have values close to 0. And then R, we see that our interaction radius increases as we get further out from the retina. But the model at that present time didn't take into account all those factors that we have within our data set. So we wanted to include as well the fact that we have a hardcore distance as well. And then also we have information about spatial covariates in terms of we have the distance from the optic nerve head. We also have the samples being taken at the 4 different quadrants really, and there is believed to be a sort of relationship in which quadrant experienced losses first, so we can look at the angle as well as a covariate. And then essentially how we worked that, or how we incorporated that into our model, was for the simulation part of the process, When we simulated a Gaussian random field, we also then simulated the log Gaussian Cox process based on these parameters and based on the spatial covariates that we had. And then as well, what we did is then applied the birth-depth algorithm based on that. And then as well, we also have then that the points can't inhibit less than a distance of r naught. That is our hardcore process. But then as well, we also wanted to incorporate the fact that the cells have size, so that maybe there's regions where cells are bigger, and those bigger cells will have a greater influence or interaction amongst each other than smaller cells. So that's why we've scaled our interaction radius based on the average of our average cell size of our mark, which is where our R over M is coming from. So when we did this, there was very little change in terms of the parameter estimates for our Gaussian random field parameters, so our mu, our sigma squared, and our S. But what we did find is that now our gamma is no longer close to 0. We have different values of it throughout the region, but that's because we've taken care of the hardcore process values, or the fact that the cells have a hardcore, really. As well, our interaction radius, as you can see from in the top and bottom quadrants, it shows there is a difference there compared to across from left to right. This actually corresponds to the density of the larger cells. So essentially, it's taken into account the fact that the larger cells are having more of an interaction with each other than the smaller cells as well. So from this, we then wanted to sort of say, well, could we sort of analyze this through time? Because of the size of the data set, applying a temporal model was very complex. So what we did was we looked at sort of some measures from change point detection instead. So really, it's looking at a cumulative sum chart. What we do is we obtain parameter values for our pattern, and then we can simulate patterns based on these. So we have multiple realizations of it and take a mean of those realizations, or sorry, get an estimate or a non-parametric estimate of the intensity for that pattern, and then apply it then for the different time points as well. And then what we do is we calculate the average of these values to see whether or not there's a change from the deviation by using the cumulative sums. So the cumulative sums are looking at essentially whether or not we go from an increasing towards the mean or decreasing away from the mean. So once we identify if there's a change point or not, what we do is we can apply what's known as the mean squared error to detect where the change points are occurring in the data set. So this is sort of trying to highlight the areas of where we find change points. Now, we focused on one retina just for this example, but essentially this is the areas where the loss is occurring. So for the 7-day model, we can see the majority of change point is happening in that smaller central retina and in the top and bottom quadrants. And then as we go along, that increases, but then also then the left and right quadrants also show a change as well in the loss of cells. So really then, just to sort of summarize, what we've done with the fractals, we're hoping to shortly have a paper out in terms of that. The measures that I mentioned, the lacunarity and succularity, they're not really used as much. So in context of mark patterns as well, we developed a code in our function to calculate these values. So the paper will have those included. As well, then the change point information will also be included in a different paper, but what I didn't include was the fact that we have replicates and we have different people. So we have that also in that paper coming as well, which we'll then look at, rather than looking at just one individual pattern across each retina, just a group pattern that can be followed through time to see whether or not a change actually does occur in the pattern. So thank you all for listening.
I think that's a tour de force. I don't know what the rest of the audience think, but that's really fascinating. I hadn't come across this box process with drugs.
Yes.
Effect.
It's a fairly recent paper. I think it was 2022. It was published. So that's why it's quite basic at the minute. So we sort of adapted it to our particular pattern, really, for looking at disease.
Is it, would you describe the whole process of fitting as easy or?
When the code works. When you get the code written down and works, it's fine. But yeah, getting to that point is, yeah, it's not too fun. But no, it's not too bad in terms of once you get it working and once the pilot runs come in and you have that value of epsilon, it doesn't take too bad in terms of computation either. It sort of helps the whole process.
I was fascinated at the beginning to know the relationship between these measures that you were presenting to us.
Yes.
Not having heard of all these additional circularity.
Yes.
Before in any great detail. I was interested to know, what is the relationship if you one person and you measure all these multiple measurements on them. Are they correlated? What?
No, they're really kind of, so if two patterns, if you look at the fractal dimension, they're different. People stop there really. But then what happens is two patterns might have the same fractal dimension. So there's no distinguishing them between their fractal dimensions. So then they look at the lacunarity. So the measure of how dispersed the image is really, or the points, in our context, the points are. So are they grouped together in big clumps, or are they maybe smaller clumps but random points through it? And then the circularity is kind of like a final measure of the lacunarity appears to be the same, the fractal dimension appears to be the same, but these measures, we know there is a difference in them, these two patterns. So then we'll look at the percolation and how essentially how the flow of fluid can pass through it, whether there's sort of a lot of gaps between the points or whether or not actually they're closed off. Or
what about the possible approach of you have guys who are normal, so you have these measures on that, okay, and then you have these measures on the guys who may be abnormal.
Yes.
Yeah. So if you try to discriminate between these two sets of data, can you detect if the guy comes in with if I come in and measure me, can you tell immediately from comparing me to what you have? Yeah,
so really what it comes into is, so for example, the value of our fractal dimension tape, for example, first of all, if your pattern is random, you will have a value of around 2. So it's actually close to the actual dimension of a square, or nearly over your study region. And that's kind of similar to what will be observed in the healthy retina. They sort of stick around a value of 1.92. But then as the disease progresses, we obviously get patches of where cells are missing, really. And then those values essentially are pulled down towards, I think it's about 1.5 from memory. And then a similar sort of thing for the circularity and the lacunarity. So circularity is the probability. Essentially, for the healthy pattern, your probability is quite low because there's a lot of cells that can't really move too far. But then as more cells are being lost, that probability shoots up. So actually the fraction of the ones that are at that infinite cluster, so as in if you pour the fluid, it can connect one side to the other. The fraction of the ones in our control group is much less than the ones in our disease group, so they're different that way.
I mean, it seems to be relatively complicated to get to you're estimating the likelihood and things like that, of rather estimating the changes through your APC algorithm. Do you see a possibility of this working dynamically? So you're outside the doctor now and you're the patient.
Yeah, so really, that's where we come in from, and using those fractal measures as summary statistics in our ABC algorithm. So the idea is that it will feed into that really. And then in terms of like the change points, the change points we have applied in the context of a neural network once we've done the replication side of things. So essentially it means that you can give a pattern into it and it'll tell you whether or not a change has occurred relative to those healthy retina. So essentially it's using what you obtain and then seeing whether or not there's a difference between what would be healthy and what isn't healthy.
In the healthy group, you have clearly a lot of variation.
Mm-hmm.
So, I mean, typically in any healthy group, they're not all have a multi-dimensional data. They're different. So is there any advantage of looking at the variability of comparing variabilities in the two samples? Is there any advantage in modeling that?
I haven't thought about that, to be honest, at present.
You've thought about enough.
Sure.
I'm sure there are questions on the
Nothing to yes.
the audience also something.
Thank you, Michelle.
Yeah, sorry, go ahead.
Absolutely fascinating. I just wonder from a diagnostic perspective, the value of this. First of all, in terms of the speed of detection as well, so will it be automated if it does implement in practice? And the confidence that you have that the point of detection will be better or worse than others at ruling out false positives. Also whether you intend to use the result singularly to say yes, no, you do or do not are a candidate for glaucoma, or whether so much of it in the context of behavioural data. And I was just wondering what your thoughts were about that. And I had just a small question that based on what Gil was saying, which is, have you thought about comparing this to aging? Because that kind of macular degeneration is similar, and the value of the quadrant analysis that you have, I wonder if there's some measures that would be very good at pointing out particularly glaucoma rather than other diseases.
Well, really, so as I said, there's recent advances, which means that clinicians can observe sort of these smaller subregions. So really it would be looking at taking it and looking at applying it to that sort of data first of all, more so than anything, because obviously we don't know the age of the mice and we only know how long they've had the disease, so we can't really work with age at the minute. But it would be really down to transitioning it and applying it to those measures to see whether or not it translates, because at the end of the day, the mice, yes, they are very similar, but they're not an exact model for human behavior. So you would need to first of all obtain data for this type of for human really, and then use it to see whether or not it actually is working in that context and apply it there in terms of whether yes or no it can be used, so whether or not you have glaucoma.
Well, I think if there are no other questions, I think we'll give that. Thank Jonathan for next one, John.